So I'm Tim Gallagher. I'm an assistant professor from Kent State University in the United States. And we're out here collecting soil gases under the spectrum thicket. Just look at respiration signals from below the ground, basically. So what we're doing is putting these steel tube gas wells into the ground. We use a drill to just spin them down. And then we have a little slit at the end where we can then put that deep underground and then connect a tube to the top of it, as you'll see in a sec, and then we can collect gas samples from beneath the surface into little gas bags where we can then take them back to the lab or to wherever we're staying in the field and analyze them for both their CO2 and their oxygen concentrations.
And what does that tell us about what's happening under the soil?
So it can tell us a bunch of different things. First and foremost, just how much respiration there is, so how much below ground carbon cycling there is, and specifically lots of people look at that CO2 value to understand, to basically quantify how much of that carbon is being respired by the plants and the microbes and fluxing back into the atmosphere. But by pairing that oxygen consumption with that, we can look at the ratio of that CO2 production to oxygen consumption, and we can start to ask questions and look for signals of different metabolisms. So is the bulk of the respiration being driven by the respiration of carbohydrates versus lipids versus organic acids versus one of the things as part of this project we've been looking in is, do we see evidence of this oxalate carbonate pathway where microbes are breaking down that oxalate, changing the pH, and precipitating calcium carbonate under the soil, which should have a really distinct gas signal in terms of that CO2-O2 ratios.
Pretty cool stuff.
Yeah. Okay. Let's just explain what.
Yeah, so you attach a tube on there,
Yes.
go around this side of the tree.
Yep. And that's where Alyssa is manually using a syringe to basically just pull back the gas sample through the bottom of this well, and then she can switch the valves around and push it into that foil gas bag that's nice and gas tight, so that we can preserve that CO2-O2 signal when we measure it later tonight back at the guest house.
So you feed that into a fairly fancy analyzer. And
it's funny. It's fairly fancy. It's incredibly, the fanciness is just them sort of coupling it. The CO2 analyzer is just a standard dual wavelength IR sensor, so nothing fancy with that. But the cool thing that this company that makes this analyzer does is they have a fuel cell oxygen sensor. So basically, the amount as that gas is flowing by the sensor, there's a diffusion gradient, and the higher the oxygen, the more oxygen passes into this electrochemical solution, creates a voltage that we can measure. And that oxygen precision, the signal on that oxygen is the really special thing, because, right, we're measuring, we want ppm, so parts per million levels of precision on that oxygen gas signal, but we're in the atmosphere, so it's sitting at like 20%. Right? So we need a really fine precision on a really large signal. So that's the trickier one. Whereas the CO2, because it's a smaller signal on a small background, is a much easier measurement to make.
Could you say anything about nitrogen fixation with this?
Potentially you can, right? Because any of those times that some of that nitrogen is affecting, some of that especially with nitrification, might have some amount of oxygen consumption.
Without CO2 release.
Yeah, exactly. So that could drive that ratio lower. It's all about the magnitude of those relative signals, right? How does that compare to the size of the respiration signal, and how do you isolate it? I think we've tried to look a couple times in the US in sort of natural sites, and we actually took an ammonia gas analyzer out there, hoping that would sort of catch some of the just natural equilibration of the ammonium into the gas, and we didn't see much. It was at the like ppb level, but we think definitely you would see it in any place where there's agricultural fertilization, or if you knew what environment and had some other indicator that you knew what was happening when with that nitrogen cycling, you could sort of look at it.
So I have an interesting theory about what happens in these soils. Is that firstly, when we have these big rainfall events, the soil's very kind of coarse here, because it's got so much litter, like broken down litter in it, and that effectively these soils become flooded, and we get incredibly high levels of bacteria
Really?
Well, the bacteria activity should go through the roof.
Yeah.
I mean, if we're just looking, there's plenty here for them to...
Plenty of food. They just need the water.
So then the conditions should become anaerobic.
Yeah.
And so then when we get anaerobic conditions, we should start getting nitrogen fixation.
We should. And you know what? We should actually talk more, because one of the new things with another young professor in the US that I've been working on trying to do is sort of customized redox probes. So we could basically, with the same sort of setup that we built at Coimbra, with the central controller that's just doing solenoids, we could basically customize the voltage that these redox probes sit, handmade redox probes, not the store-bought ones, and sit right at that sort of ideal voltage signal for something like a nitrogen oxidizer reduction.
Because that could also explain that we're sitting in a site where it gets 290 mils of rain or less a year, and yet we have a closed canopy. We're sitting in a bush club, but this whole area should be a closed canopy forest. And part of the thinking around that is, firstly, it's the amazing system altering properties of specboom, but it's also the potential for nitrogen fixation, which is incredibly rare in semi-arid systems, like rare to entirely absent. So to have a system that gets these spikes every time it rains, we get a spike in nitrogen fixation, which would explain the biomass here and the higher, slower productivity, but the fact that that like this amount of plant material can be supported.
Totally. No, it's fascinating. It's fascinating.
Cool.
Yeah.
I'll leave it there.
Yeah.
Thanks.
Yeah.